Um, okay, so keep posting questions. I've seen some questions come through the chat, some through the Q&A, some through the Slack, and some through email. So I'm going to just start at the top and work my way down, but feel free to keep asking questions. We have about 20 minutes, and then we will take a quick 15-minute break. So the first question came from Dr. Jen during Ken's talk, and maybe some of this got answered, but Ken and Yui, the question is about the accuracy of the pH sensor in the QC process. We get this question a lot, and Yui, I know that you got to highlight this a little bit, but do you want to talk a little bit more about how you know what the accuracy is and what you think it is, and how you get rid of bad data?
Yeah, and then so yeah, kind of quickly walk through the process for QCing, but so we use a deep reference pH field calculated from empirical algorithms to assess drift, and also just any calibration offsets. And then so just based on global, GLODAP data, those algorithms we think is accurate about 0.007. And so, so the sensor data can't be any more accurate than that, because that's how we adjust our data. So that's kind of the bottom line. And then so, so based on the validation of the accuracy really comes from our bottle data, which really does suggest the accuracy around 0.007. But typically, I think we are a little bit more conservative and just say that pH sensors are accurate to 0.01. That's kind of the value that we report in papers typically. And so, yeah, so that's and then QC process, just spike, spike detection, just looking at the data. If it looks weird, if it starts drifting, there are some diagnostic measurements from the sensor itself that we can use to flag that data. Yeah.
Great.
Yeah.
Go ahead again.
I would just amplify, I believe it's shown in Tanya and or Yui's extended talks, but we also look at crossovers with GLODAP data. As the floats go near historical GLODAP stations within a 20-kilometer distance, we compare the deep, typically below 300 meters to get rid of seasonal effects, but look at the deeper data, and that also supports, the idea that there's very little that these initial estimates for pH of order of 0.01, 0.007, for example, or nitrate, 0.5 or so micromolar, are sustained throughout the life of a float. So beyond the initial estimate at float launch, we are looking also throughout the float lifetime.
Great, thanks. The next question. Oh, go ahead.
Oh, I just wanted to add, so those float to bottle matchups really help us determine the sensor accuracy too. And there's always assumed that there's a bit of that error is in the spatial temporal mismatch float to the bottle data. But that's also why we like to perform that analysis routinely and see how if we're getting better over time with an improved sensor development. So that's a key. The repeat analysis is a key part of that too.
Great, thank you all. The next question is for Steve. So this question comes up a lot where we think about the longevity of the floats versus how fast we need to profile them. And you kind of quoted this 10-day cycle that they will live for a pretty long time based on the energy budget. Can you say anything else about the possibility of cycling these floats faster?
Oop.
Oh, you're still muted.
Okay. The energy budget, as I showed, says something like 260 profiles. And that's the budget if the float fails due to its batteries. But there are other things that can take floats out. Sometimes the sensor fails, we get the occasional corrosion, and then there are a lot of things that we don't know, but that on a good day, if everything's working, about half the floats will make it to 260 profiles. So we really need the floats to work for 4 years, which means a lot of them have to work, if that's the average, more than 4 years. It's possible we could cycle faster. And I think once GO-BGC gets going and we have a lot of floats in the water, we can see how we can do with that. Maybe we could come down to 7 days. But the idea of cycling at 1 or 2 days is probably not practical. We could not maintain a 500 float array that way. So From 10 to 7 eventually might be possible. I don't think we can go any faster than that and hope to maintain a 500 float array or a 1,000 float array with the rest of the world's contribution.
Thanks.
I want to add to that too. So like daily cycles for, so one of the reasons why sensors are so stable on floats is that they live the majority of life in a refrigerator, right, under high pressure. And then so if you start cycling every day, then you run the risk of biofouling your sensors as well. So then you also compromise the sensor, the performance as well, So and there's evidence of this in the Argo data center where floats of oxygen sensors that come up during the day go up to like 500, 600 micromole, and they don't do that at nighttime. So that's a clear sign of biofouling. So that's definitely, we can't compromise.
I might add to one other thing to that, and that is there are certainly good reasons you might want to cycle faster. And as I said, we're probably not going to be able to do that. But for people who find that that's a requirement for their research, we're going to hear a couple of days from now from agencies, at least in the US, who may entertain proposals to do other things besides GO-BGC with floats like this. So there are other possibilities, not necessarily with GO-BGC, but with similar floats to do other kinds of missions.
Thanks. Yeah, I mean, just following up on that, the mission here is kind of this long-term observing system. So we're kind of looking at long-term stability and keeping the floats out as long as possible. But like Steve said, there's a lot of reasons to cycle faster. Okay, next question was for Yui. And the question was about, you mentioned having different sensor types. And the question is, does using different sensors on different floats cause a problem? And it was specifically, I think, about you mentioned about oxygen, but it's true for a number of the sensors that there are small tweaks in different sensors.
So it does create a huge problem in the data management side, as Tanya knows, because we have to keep track of all of the metadata and different coefficients and all of that. But most of these sensors, so pH, for example, we have gone through very thorough interlaboratory calibration efforts to make sure that the sensor performance is basically the same between Seabird and MBARI. Same thing for the nitrate sensors, and these results have been validated in the field as well through, so the pH and nitrate comparisons that I've showed is a combination of both the Seabird and the MBARI sensor types for pH and nitrate, and then so we don't really see a significant difference between the two. So there's so the answer is no. Like we have done our best to make sure that these sensors are comparable, and the basic sensing technology is the same. The core components are the same as well, so it makes sense that they would be comparable. The oxygen one is the key difference. These air calibrations are really important to obtain the highest accuracy for oxygen. So if you air calibrate them, you can get oxygen accuracy to about 1%, but if you don't, for the Seabird ones, we rely on either calibrating to Winkler samples at the time of deployment if they're available, or comparing it to climatologies, which then the accuracy reduces down to about 2 or 3%. And we also can't assess for sensor drift over time. So that's the main difference. But we're, there's a funded proposal to try to improve this, to kind of fix that problem. So having a Seabird sensor that can do air calibration and also benefit from the pumped faster flow rate. And so those are looking very promising. So we anticipate those would be integrated onto our GO-BGC floats in the near future.
Yes, Ken?
Yeah, I just wanted to add a comment though that GO-BGC, BGC Argo, the technology can't completely stay frozen in time and always using the same sensor. So as an example, oxygen on floats originally started with a Clark, the classical Clark electrochemical sensors was supplanted by the optode optical oxygen sensors. They just performed better. And, we will have to, and it causes big problems in the data system for sure when you try and compare the older Clark's data to the newer optode data. But I think that we do have to, retain the ability to adapt. Otherwise, that causes an equally big problem if we, never change sensor types.
Great. Okay, so the next question is, oh, and I would like to remind everybody, you also have the option of just raising your hand, and that's under the reactions. If you look down the bottom, there's a smiley face plus, and you click on that, there should be an option to raise your hand. So I can keep reading people's questions, or you should feel free to raise your hand. Okay, so the next question is, would multiple spectral fluorescence be an option at some point for integrating onto floats? And this wasn't directed at anybody, so who wants to take this question?
So the answer almost follows from my last comment that we do, you know, have to improve. There are issues. The fluorometers and floats now, you know, excite the sample at one wavelength and look at emission at another. And there is a lot of value to looking at multispectral excitation emission. There is a project underway out of Hervé Claustra's lab to start looking at fluorometers that excite at just 2 wavelengths. And you start to learn something about the plankton species and so on. So I would expect that as time goes and as optical technology gets better in the future, that we do start to look at more exotic, you know, chlorophyll fluorometers that are capable of, you know, multi-wavelength excitation, multi-wavelength emission, just part of the general improvement. The the flip side is that a program like GO-BGC, funded for 5 years, we wrote a budget based on single wavelength fluorometers. So, you know, adapting to new technology just becomes a challenge from a budget perspective.
Very true. Any other comments? I think you
Yeah,
did there. Oh, go ahead.
This is Eric Green from Seabird Scientific. I'm the scientist who's working with Hervé on the multispectral fluorometers. And I just wanted to point out, since this is a GO-BG seminar or workshop, you know, sort of what the motivation is, and it's twofold. And you can find this, you know, amply discussed in the literature. But the reason we're starting with 1 wavelength is that we're looking to reduce that uncertainty that was shown earlier by Yui with respect to that global average of almost a factor of 4, where the global average was looked at as essentially a factor of 2 too high. The idea is that by for example, more directly exciting the chlorophyll A pigments with a different wavelength, we can reduce that variability. Of course, that requires global measurements to validate and some time. The second is, and again, you can also find this in the literature, that multispectral response can help us with community composition. And more wavelengths is traditionally better with respect to the work that's been done in multispectral fluorometry, whether that's in situ fluorometers or, you know, flow through or even laboratory work. But that's the direction we're going in. So there are essentially 2, you know, sort of possible wins with multispectral fluorometers. Thank you.
Yeah, thank you, Eric, for adding that. Okay, our next question kind of follows on this. Is there a pathway to more quantitative chlorophyll measurements than just fluorescence?
You know, I think that there is a lot. So one of the sensors that we have not used a lot in the US, but we are moving as fast as we can towards incorporating into the array, are radiometers. And Josh Plant, who one of the data analysts for SOCOM, has been looking a lot at a comparison of, and Christina Schallenberg, I think, probably on as well, and maybe going to talk about this tomorrow. There seems to be a pathway towards combining, and has been shown in Hervé Claustra's group, Xiaogang Xing. Merging radiometry and fluorescence seems to be a way to make this also, the whole chlorophyll number, more quantitative. So there is active effort. GO-BGC intends to deploy radiometers, and we hope that will make the chlorophyll numbers much more quantitative.
Thanks. The next question is from Tom Mitchell, and I'm going to kind of con people. He has a longer question here. Tom, if you have a mic, can you just ask your question? If not, I can read
Sure,
it.
thanks. It was just about uncertainty and how it's handled within the database. So we have some derived products such as PCO2. It's derived from two different products or two different measurements. And when you're doing your analysis, can you backtrack to figure out what the major contributor to uncertainty was in your derived product versus, hey, they're both equally weighted? I'm curious how we're tracking uncertainties through the data flow.
I guess I can take this one. So we have done a substantial amount of work to try to quantify the uncertainties associated with the derived carbon dioxide parameters, particularly PCO2, because that is a particularly sensitive and finicky parameter to derive. But so Nancy Williams has a very great paper, 2017, that looks at a very detailed bottom-up analysis, uncertainty analysis on what contributes to the overall uncertainty. And she did the same kind of thing for DIC and aragonite saturation state. I have a paper using glider pH data to do a similar approach for using Monte Carlo analysis. So we have a pretty good understanding on what contributes mainly to the uncertainty. So in summary, like DIC is basically an uncertainty is basically equivalent to the estimated alkalinity. So that's a dominant term. But for saturation state and PCO2, it mainly comes from the accuracy of the PCO2 pH sensor itself. And then so the derived products aren't served on the Argo dataset, but only through the MBARI the float viz portals and our snapshots that's hosted by Scripps. And then so for those, we do claim, there are associated uncertainties with those variables that are based on these papers that we have written. And yeah, so I go into this a little bit into our into the curated talks, but I'm more than happy to send some of these papers too. And I kind of want to make one plug again. So Ken and I have actually a funded project right now to try to reduce uncertainty in the PCO2 derived from pH sensors. And so we're hiring a postdoc for that. So if anyone is interested in that or know anyone, who wants to solve this problem, because it is a huge problem moving forward, is trying to get global CO2 estimates is one of a very important application for GO-BGC. So we want to get to the bottom of what's causing some of this uncertainty.
And maybe just to follow up, Yui, I think one of my suggestions is, as GO-BGC is trying to introduce new users in the community to the data set, to have a way of really accurately describing uncertainties and how they come through in different measurements, it would be helpful that they don't have to know, well, I go to this paper, I go to that paper. I mean, it is one where somebody who's been like yourself in the field for a decade or more, you kind of know which ones to go to. But I think for new users, it may be very helpful to understand, this measurement PCO2 is pretty tweaky and there's a lot of assumptions that are built in. And then there's others like temperature where you can bet that that third decimal place is good, and it might even be a fourth decimal place out there that's very stable. But just understanding where, for new users, where the uncertainties come from and how to address them. Thanks.
Yeah. The next question is for Steve, and I can kind of tack on to the end of this. It is, can you talk a little bit about the vertical sampling resolution and if that will be different for the solo float? Steve, do you want to touch a little bit about how, what the vertical resolution of sampling is right now?
Well, for temperature and salinity, we're sampling at 1 hertz, which gives you a sample at about every 8 centimeters, and that's too much data to send back. So it's binned into 2 meter bins, and that's still pretty good for almost anything we need to do. For the BGC variables, we collect about 100 points of each one, 100 samples. And so near the surface, they're 5 meters apart, and as you get a little deeper, 10 meters apart, and a little deeper 20, and when you get into the deep water, they're 50 meters apart. But altogether, there are about 100 of those. And this is all dictated by how much data can we actually send back. And right now, if you look at the talk that's the full version of my talk, I really get into that in more detail. But Right now, we're sending about between 50 and 100 kilobytes of data per profile. And if we were to sample more often, we